Viruses. What are viruses? A virus is a minuscule infectious particle which replicates inside the living cells of an organism. A virus is not considered living because it lacks many of the necessary characteristics. These characteristics are reproduction, adaptations, reaction, energy, homeostasis, cell growth, development, and lastly, genetic code. They can also infect all types of life forms. This includes, but is not limited to, animals, plants, fungi, and even bacteria. Viruses are also roughly grouped into several main categories. First is helical. These are composed of a single type of capsomere or the subunit of a capsid stacked around in a helical structure. The DNA or mRNA is stacked in between the structure or is bound to its proteins. The next type is icosahedral, or an animal virus. The capsid of the virus is again made of capsomeres and proteins stacked in a triangular structure with 20 different faces and its nucleic acid inside are intertwined. The last is complex. These viruses are commonly bacteriophages, meaning they infect bacteria and are composed of protein tails, complex outer walls, a capsid head, and a shield on a base plate hiding a pin used to poke the cell membrane. An important structure is the virus envelope. This membrane is composed of an outer lipid bilayer known as the viral envelope, and the membrane is studded with proteins. It also has lipids and carbs which are taken from the host for nutrients and for its membrane. The last main structure is the virus genome. This genome is the DNA or RNA. There are millions of types of viruses, more than any other group like plants, animals, and bacteria, and only about 5,000 have been listed in any sort of detail. All viruses have genetic material, a genome, made of nucleic acid which code it. Viruses can have possible combinations of their strands of nucleic acid type. They can also use the same genetic code as their living host cells, so they can reprogram them for reproduction, similar to recombinant DNA. Viruses cannot asexually reproduce and are acellular, so they must use other cells to reproduce. First, the virus will attach using its protein layer and lock onto the organism's receptors. Next, the virions will enter the cell by piercing and passing through the cell's membrane. Then, once inside, the virus capsid is removed and the nucleic acid is released. The nucleic acid will take over the host cell, cause rapid replication coded for new viruses, where the cell will overload, explode, and infect many other hosts or cells. There are two virus replication cycles within the host cell. The first is a lytic cycle, where the virus is active and is constantly replicating. The second is lysogenic, where the virus is dormant. Once certain conditions are present, the virus will become lytic again and replicate. During virus replication, genetic mutations can also occur. The first type is antigenetic drift, where DNA or RNA base is changed, a type of point mutation. Next is antigenic shift, where there is a major change in the virus. It can possibly change the entire nucleic acid sequence. This can be a result of recombinant nucleic acid. This is where the virus breaks a strand of nucleic acid in the host cell and replaces it with its own. These genetic mutations are what causes viruses to be able to change over time and grow more resistant or immune to vaccines. This commonly occurs in influenza or the flu, and is why we require a new vaccine every year to stay protected. Some common viruses include the flu, the cold, rabies, measles, Ebola, HIV, polio, and more, like listed. Lastly, my article was about viruses that cause cancer and tumor growth within humans. They are known as oncoviruses and can be either DNA or RNA viruses. They work together to cause cancer with other cells, causing factors such as air pollution, smoking, etc., and together will attack your living cells, such as tumor suppressors and cell regulatory cells, which cause abnormal growth of the cells. DNA viruses can cause cancer in many ways. The first is integration, where they insert their own DNA causing mutations. Second is between certain checkpoints of cell replication and can cause premature or too much replication. Third is by turning off the p53 gene, which is a tumor suppressor, and last is the RB gene, which is also a tumor suppressor, but prevents too much cell replication. Another type is RNA, oncovirus. These viruses create a double-stranded DNA copy within of the RNA within the host cell. They then reverse the sequence and insert it back into the chromosomal DNA. This can have very negative side effects because the entire strain is affected. Some examples are HPV, herpes, hepatitis, and T-cell. Hopefully, through more research and studies, we may be able to better prevent these viruses and reduce the amount of cancer cases in patients. That's all. Thanks.